Done. So as we move into producing protein, we have two major processes. Our two transcription and translation. So we're going to start with transcription. And remember, transcription is taking our DNA and what are we going to produce from our DNA? RNA. Same language, same alphabet, nucleotides. We're just going from DNA to RNA. And the principal RNA that we think about is our messenger RNA. That's the code we're going to use to know how to assemble our protein. And that's going to be how do we assemble the amino acids into the correct primary sequence to form a functional protein. Now, this is our genetic code, this wheel that you see. It has our amino acids, and our genetic code are three-letter words. And I think you're pretty familiar with that already. We call them codons. And so we're going to be talking quite a bit about the significance and the actual language of our genetic code that we can use 64 combinations of our 4 nucleotides into these 3 words that encodes for our amino acids. That's a big part of understanding how transcription and then translation is going to occur. So, we talked about DNA. That's just making a copy of your code. That happens in the S phase of interphase. So that's just DNA replication. But we've already said DNA to RNA, transcription, and then chapter 22 is going to be translation, taking that code and assembling the amino acids. So we're pretty good with the difference between DNA and RNA, right? What's the big difference? What do you find in RNA that you don't find in DNA? Uracil. What do you find in DNA that you don't find in RNA? Thymine. That's sort of a bit of a tricky part. The other differences we have are our RNA, single-stranded, whereas DNA is double-stranded. And then, of course, the base sugar that we use is ribose versus deoxyribose. Now, RNA production, our transcription from our DNA, in many ways is a much simpler process. But when we look at our DNA strand, we have 2 strands, right? A parallel and anti-parallel strand. We're always thinking 5' to 3'. But when we look at our strands, we have to understand that one of our strands, and in this particular instance, it's a 5' to 3' strand in black, that is what we call the coding strand, or the sense strand. That's the code. You read that code, you know what the amino acids are going to be. But our anti-parallel strand, we call the template strand, because that's what our RNA polymerase enzyme is going to use as a template as it assembles that strand of RNA. And the RNA strand that's assembled is going to look in code exactly like the coding strand of our DNA, with one exception, the U's instead of the T's. So here we see TCCAAT, UCCAAU. So do you see how they're identical? The messenger RNA is identical to the coding strand of the DNA, with the exception of T's in DNA and the uracils, the U's in RNA. But what RNA polymerase is going to read is it's going to read our template strand so that it can produce that RNA strand that has the same code. There's another reason why it uses the template strand. How do you assemble a strand of DNA or RNA? It's the same. In what direction do you assemble them? 5' to 3'. So if you're going to assemble them 5' to 3', automatically you know what direction are you going to have to read the template from. The opposite, 3' to 5'. So that's another really logical means in which we have to use that template strand to be able to assemble that coding strand into our messenger RNA. So now the genetic code. This is the big part of understanding transcription. When we look at a gene, and we'll just say that here's our messenger RNA, the same as our coding strand of a particular gene, 5' to 3'. We divide our nucleotides up into 3 letters. Those 3 letters representing the codons. When we get to translation, we're going to see that each of our 3 letter words will signify a particular amino acid. And we'll go ahead and introduce the concept AUG that you see right on the far left, right by the 5'. AUG is the first codon in all proteins. It is called the start codon. That encodes for the amino acid methionine. Methionine, M-E-T-M, it's the first amino acid in all proteins. Because you can't have a protein unless you start it. You can't start it without AUG, which is the one and only codon for methionine. So methionine has to be first. So you encode for methionine. Here's a GGC, which is glycine. Here's a UCC, which is serine. You see how we are translating the nucleotide code into amino acids? Completely different language. But we first have to understand the code, these 3-letter words, and how they then are assembled and what the significance of these are. So if you have 4 nucleotides and you assemble them into 3-letter words, that means there's 64 combinations. How many amino acids do we have? 20. So there are going to be some that more than one codon can encode for those amino acids. Methionine has only one. There's another one that has only one, and I'm drawing a blank on which one it is. But then we also have 3 codons that are going to say stop. It's as important to know where to start building a protein as well as knowing when to stop assembling amino acids and the protein is done. Possibly, I can't remember. It'll be coming up in one of the slides. I can't remember before we get there. So when we look at a couple of ways to place the right code with the right amino acid, this is what we had coming up. We had on the left-hand column, we had your first nucleotide in your codon, U, C, A, and G reading down. Across the top, you had the second codon, U, C, A, and G reading right to left. So in the upper left-hand box, you can see our codon is U, U, and then the U and the C are the codons for phenylalanine, and the UUA and the UUG, those are codons for leucine. And so on and so on and so on. Here's our AUG in the A box for the first codon, in the U box for the second codon, and there we have the G sequence. That's your methionine start. Now we also have our 3 stop codons. Now I wish it was easier to remember those. I only really remember one, and it's this one, UAG. What is the coding sequence in DNA? It's not UAG, it's what? TAG, which is tag, and to me tag means stop, freeze tag. When you tag somebody, they stop. That's how I remember that one stop codon. Most of these you just have to commit to memory. And you're going to be asked codons and amino acids. But I've spent most of the morning trying to simplify that ordeal because it's a task to remember all these. Now, this is sort of a new way of trying to help you remember the codons for the amino acids. The first codon is in the center. The second codon is in the next concentric circle coming out, and the third codon is the next to last concentric circle, and then you get the names of your amino acids, as well as your 3 stop codons. So I thought, okay, well maybe if we color coded, because I'm a visual person, and maybe if we color coded it. So I sat down with Photoshop this morning, and I said, okay, if we do red, yellow, blue, and green for our primary, nucleotides. And here where you have U and C, if you take red and blue and put it together, that makes orange. So instead of having to remember U or C, if you remember orange and you remember your primary colors, you might get that. Here we have red and blue that gives us this sort of, this magenta type color, cyan, I think that's what that's called. And then we have one that are 3 A, C, and U, 3 amino acids that give a brown color. And if you can use all 4 nucleotides, or any of the 4, then you get, those are the black blocks. I don't know if that simplified it or not. So I looked at it and I was like, that still looks busy. Okay, maybe there's a better way to do this. So as I was looking on Google, I found what is called the consolidated scheme, or the combination scheme. So, and I tried putting these on white, but that background didn't show up. So let's look here for alanine. We have the G, guanine, and it's neat, G is green. I accidentally did that, and I was like, oh, that's cool. C, cytosine, being yellow. And then N, which means any of the 4, is in black or in gray. So if you could use an A or a G, that's going to be that cyan color. U or a C is orange. Here's ACU, that's going to be the brown. And then here are our stop codons. Methionine, I made green and underlined it so you would remember that's a start codon. That's still complicated, right? But you see how I'm trying to simplify it more and more? Now, if you remember these letters, and you remember what R is and what Y is, and what the H is, simplifying it even more. But I thought, well, maybe there's an even better way to organize it. And so what I did was I took our list that really just, it's a list of alphabetical amino acids, and I said, maybe that's not the best way to remember codons with the amino acids. And so I made this list. So I broke it down. Here are the amino acids that all end with the N, any of the 4. Here we have all the amino acids that end with the Y, which is the U or C. Here we have 3 that end with the R, which is A and the G. Here is isoleucine. That's the one that's got the H on the end, and then the tryptophan. That's the one that's got the single codon, much like methionine has a single codon. And then we have 3 amino acids that really are complicated. They have sort of 2 separate ones for each. So do you see how, as you organize the information, regardless of how it might be presented, it becomes a little bit easier, a little bit more meaningful. But see, even for me, I looked at that and I was like, well, maybe I can make it even more simple. So what I did was, especially like for here, I said, well, they all end in N, so I don't need to write that one. And up here, we've got these 3 that end in C. Here are these 4 that end in A. So I said, okay, these all end in N. I don't even need to really put that in there. I know the second codon for these 3 is a C. The second codon for these 4 is an A. All of these have the AR. So now you just have to remember Q and C, K and A, E and G that are the ARs. So you see how as you step it down, you organize it so it's easier to remember. Now, this is the way I tried to put it together. Whatever is meaningful to you, whether it's the colors, or if you like different colors or a different organization, this is how you can remember which amino acids belong with which codon. but to me the two most important are methionine with the AUG, the start, and the stop codons, of which UAG, TAG in my mind, tag, means stop. Any questions? Okay, this is sort of a new PowerPoint that I put together, so I'll make sure to upload that PowerPoint, and of course the podcast will be there as well. Cancer is a horrible thing. I don't think anyone would disagree. But what must you have occur in a cell to get a tumor that might lead to cancer? A mutation. And we know various things can cause mutations. Environmental factors, radiation, carcinogens, or just random happenstance of replicating DNA. If that DNA polymerase, even though it makes very few mistakes, if it makes one in exactly the wrong place, that can happen. So as we look at our mutations, as you have your alphabet, and you're putting those words together, methionine, tryptophan, glycine, glycine is our example in this illustration on the left. If for some reason you inserted one additional nucleotide, do you see how this can mess up the whole thing? Because now instead of methionine, tryptophan, glycine, glycine, because of that one insertion, we have shifted what is referred to as the reading frame. Because you're reading every 3 letters. Well, now by inserting a C where there was not supposed to be a C, now you got methionine, cysteine, alanine, arginine, and it just happens that you get back on track here. So one insertion or one deletion can lead to a completely new set of amino acids being assembled. So in this case, we're just talking about a single insertion. But understand, a single deletion can do the same thing. You can have double mutants, you can have triple mutants. And this illustration for frame shift mutations, I like how they've done that, because look at this. They're using just our normal alphabet, and they're organizing it into several 3-letter words. And normally, if you were to take what they consider to be this gene and spread it out into 3 letters, it would read, "Our big red dog bit the old man." So imagine these are our codons. Well, if you put an X in between our and big, now it reads, "Our zibby gree do gibby." It's nonsensical. It doesn't make sense. Likewise, if you take out the G, if you deleted one, now our bur ed ogd, nonsense, right? If you do double mutations, you either add 2 or subtract 2, you're going to get that gibberish again. But notice something interesting. If you add 1, but you also subtract 1, depending on where you do that, you're going to shift the reading frame between those mutations. So in this case, we've got one amino acid that would be different, and then we get back on track. But one change in one amino acid is all it might take. You ever heard of sickle cell anemia? One amino acid different, one nucleotide different, and look at what happens. So triple mutations, if you have 3 added or 3 deleted, again, you're going to change the amino acids encoded between where you do that, but then you'll get back into your reading frame, but unfortunately, that may be enough to cause the damage. So do you understand how the mutations happen? And the numbering, whatever it does to your reading frame, that's the actual consequence of what's going on, and the consequence of possibly getting mutations. So we've got the code, we've set the stage. Now let's actually make some RNA. And we're going to make more than one type. Our code that's going to be used to assemble proteins is our messenger RNA. But we're not going to be able to build the protein without the other two types, which of course we understand are our ribosomes. That's where translation is going to happen. The ribosomes are the warehouse inside of which we're going to assemble the amino acids. But we also have these little transfer molecules called transfer RNA, which I like to think of as the workers at the warehouse that are actually going to go find the individual amino acids, bring them to the ribosome, and assemble them in the correct order. And that order being dictated by the messenger RNA. You see how you're not going to have a protein made without all three types of your RNA. So, here's the big picture. We've got our double helical molecule. Our RNA polymerase is only going to attach and start the process when it binds to a promoter region of the gene. That promoter region is also going to bind factors, other proteins that are called, guess what? Transcription factors. Guess what these things are going to be important for? Bringing in the RNA polymerase, telling the RNA polymerase where to start. But RNA polymerase is not going to need helicase. It on its own can read, produce the new template, and unzip the DNA as it goes. So you don't need all of those other helicases and ligases and primases. RNA polymerase can just sit down because of the transcription factors. Once it gets some ATP, which is the on signal, it starts reading and making RNA, sorry. Because this is RNA polymerase. So we're going to have the binding, which is dictated by those transcription factors, initiation, which is made possible by ATP, elongation, which is just continuing to produce some, and then termination of the RNA. Those are the four basic steps in our generalized view of what transcription is all about. Once again, let me point out, in our template strand, which is 3' to 5', that's the direction polymerase is going to move along the template, so it can assemble the messenger RNA molecule 5' to 3', because that's the only orientation with which we can assemble nucleotides. Now, replication, DNA replication, that happens in prokaryotic cells and in eukaryotic cells. And we first got an understanding of how this works in bacteria. As we look at a particular gene, and again, we're talking 5' to 3' on the coding strand, we know that there is going to be a start point for our transcription. That start point is not necessarily going to be our methionine codon, but it's going to at some point include that methionine for where we start assembling our first amino acid. But as we look at points that are further 5' from our start point, and we call that upstream, because if we're going 5' to 3', think of a river flowing. And if you're in the middle of a river and you start paddling against the river, you're going upstream, all right? So we refer to these as upstream regions. And what we find in our bacteria are these promoter regions. About 10 sequences away from the start point, we have these sequences of TATAAT. Those are referred to as the Tata box. That's fairly conserved, fairly consistent in bacteria. And then at about 35 nucleotides upstream, we have another promoter sequence that's not really as conserved as your Tata. Both of these are going to be important footprints on which the RNA polymerase will bind to start producing the RNA. So RNA polymerase is going to bind to those promoter regions. It has a footprint of about 30 base pairs. So you understand the significance of 35 bases upstream, 10. It's sort of within that footprint to get set down, then move up to, have you ever seen a drag race? You know, drag race, you got the car back behind the line, and after it heats its tires up, then it eases up to the line, because as soon as they get those front tires on that line, the light comes on and they take off. RNA polymerase hits those promoter sites, and then it eases up to that first start like a dragster, and once it gets to that first start, it takes off. So binding to those promoter sites are critical to produce our single-stranded RNA. Notice there's our 3' to 5' template strand of DNA. And you see our 5' to 3' DNA is being extruded out as a single-stranded molecule outside of RNA polymerase. RNA polymerase unwinds the DNA, and what happens is RNA polymerase is finished with it. It rewinds it back together. The RNA does not stay bound once it's done with the production of the RNA molecule. Now, just like it's important for DNA polymerase not to make mistakes, it's critical for RNA polymerase not to make mistakes either. So you do have proofreading capability for our RNA polymerase. Because if you've got pristine DNA, but the RNA polymerase makes a mistake, the protein's going to be jacked up. So all of our polymerases must have that proofreading capability. So for our prokaryotic cells, we've got our promoters that say where to start. Where we stop is when we hit regions that are rich in the GC bases, and then we get to a region that's got a lot of U's in it. Now, first of all, what is a region that is rich in GC? What is that going to confer to our DNA strand versus ATs? Stronger bonds. So it's almost like the RNA polymerase is going to hit that and hit quicksand. It's going to slow down because it's harder to break those bonds. And then it hits that stretch of U. That's when RNA polymerase says, okay, you know what? I hit the quicksand. Now I've hit the groove pavement, so I'm done. I quit. That's the termination signal. We also have another molecule, Rho, that is thought to bind to certain of these sequences, and that's also going to act as sort of a traffic guard that says, okay, stop, you're done, get out of here. So in prokaryotic cells, we have that means with which we can start with our promoters, the ta and the negative 35, but those GCs and U's as well as that termination factor Rho all stop the transcription in our bacteria. Now that's going to be slightly different than what we're going to see in eukaryotic cells, but what's going to be hugely different between bacteria and eukaryotic cells, our cells for instance, in our bacterial organisms, they produce a messenger RNA that is referred to as polycistronic. It's a long messenger RNA that has a number of different genes within it. So think of what happens, that messenger RNA for bacteria, it's a copy of an entire book with a number of different chapters and a number of different pages. So then you're going to produce the proteins, and that regulation of which sequence do I encode into which proteins, that's regulated at the ribosome. For eukaryotic cells, mammalian cells, you make the messages one copy of one page from one book. And that whole RNA is going to be one protein, monocistronic. So that's a huge difference between prokaryotes and eukaryotes. Pro starts with a P, poly starts with a P as well. That's how you can keep those together. Now, eukaryotic transcription, we're also going to have upstream promoter sites. But the thing we spend more time looking about are our transcription factors. And our transcription factors are proteins that are going to bind to our DNA And those interactions are going to enhance the ability of our RNA polymerase to come in, sit, and initiate, bind to the DNA and initiate and elongate our along the template strand to produce our RNA. So with DNA replication, we had two DNA polymerases, right? Which DNA polymerase was responsible for synthesizing the majority of our new DNA strand? 3. Our other polymerase that was able to come in, clip out the RNA primer, fill that back in with DNA was DNA polymerase who? 1. So we had 2. For eukaryotic cells, we have 3 RNA polymerases. And these 3 polymerases are going to be unique for what type of RNA they produce. So RNA polymerase 1 is going to synthesize the ribosomal RNA. I don't know how to link all these together. You can't do it alphabetically. You can't do it because which is most important, because you can't make protein without all 3 of these. But RNA, our RNA is produced by 1. RNA polymerase 2 produces our messenger RNA, which has the code. And then polymerase 3 encodes for our transfer RNAs. How you make your little mnemonic to remember those, I've not been able to find one that really works. I wish that 1 encoded for M and 2 encoded for R, then it would be alphabetical. 1, 2, and 3 sequence, but it's not. Now M is in the middle, and 2 is in the middle, and R comes before T. Would that work? It's kind of complicated, but if you think of a better one, let me know. So, as we said, much like we find in our prokaryotic genes, we have those upstream promoter regions upstream of the gene. And it's going to be in these upstream regions that we have a number of transcription factors. And many of them are labeled with Roman numerals and alphanumerics. It's not important that we know all of these names, and I think every day there's new ones that are identified. But the transcription factors bind to those promoter regions of our gene, and then here comes our RNA polymerase. It's also going to be associated with a transcription factor. When it comes in and it docks, we have this whole conglomeration of proteins that enables it to bind. And then for elongation, what do we have to use? ATP. You see that some of our transcription factors are going to be able to phosphorylate our RNA polymerase, and once it gets phosphorylated, boom. Now we start our elongation, the production of our RNA strand using our DNA template. So we're elongating, we bound to the DNA, we initiated the sequence because of ATP, now we elongate our RNA strand, and the last thing we have to do is terminate. And so when we get down in our 3' end of our gene, we're going to see that the RNA gets cut. RNA polymerase cuts it off from the DNA, and it's going to have about 10 to 35 nucleotides after we hit this AAUAA sequence. That sequence is the stop. It's like the finish line at the drag race again. I keep using that analogy. But the dragsters, they don't hit the line and stop instantly, do they? No, they pop the chute, and it takes them a little while to stop. Well, RNA polymerase is the same way. It's not going to stop immediately. In fact, when it hits AAUAA, that next 10 to 35 nucleotides are just going to be A's. It's like the tires screeching along the road. It's just A, and we refer to that as poly A tail. And so the poly A tail is going to be there not just as, okay, I couldn't stop fast enough, but it's actually protection against degrading. And so in fact, our post-transcriptional modification, do you see how we get that name? Post, meaning after, transcription, we're going to modify the messenger RNA, we're going to do a couple of things. That poly A tail is going to be one of them. So first, chemical modifications. So we've got these first couple of sequences, especially like of our transfer RNA. We've got these first couple of sequences that are important for helping attain a secondary structure. Our primary structure is just going to be our nucleotides. But you see, with our primary structure, we're going to create these hairpins. And these hairpins are going to form hydrogen bonds at very specific regions. And these leader sequences are going to be important for helping get that lined up in the right place. But we don't need them for the normal functioning of a molecule. And so we're going to clip off that part. So part of our modification is clipping it off. We're also going to have some nucleotides that undergo a process called methylation. And so we're going to replace a hydrogen with a methyl group. And again, that's going to prevent some nucleases from being able to come in, recognize the end of our nucleic acid strand, so these exonucleases, and they won't be able to degrade the DNA. So it's protecting. And we refer to that methylation for our tRNA, but we also at other RNA, so our messenger RNA, for instance, that 5' end is going to have a 5' cap. So the cap on the guanosine, that is going to be where we methylate that region. That's going to prevent degradation from the 5' end. And the poly A tail, again, once we hit the AAU, AAA, now we might have 50 to 250 adenines added. So it's just this huge strand. So as the exonuclease might come in, if it starts cutting off from the end, it's going to have a lot of RNA to eat before it gets to the good stuff, the important stuff. So again, that's the protection at our 3' end. The methylation is the protection at our 5' end. Now we said the difference between prokaryotic and eukaryotic mRNA was the polycistronic for prokaryotes, monocistronic for eukaryotes. Well, there's another difference. In our prokaryotes, the messenger RNA, all of the nucleotides have significance. But in eukaryotic messenger RNA, not all the nucleotides we produce and assemble into our RNA are important. We have junk pieces that are situated between the important pieces. And those junk pieces are referred to as introns. And so for our introns, we have to undergo something that is called splicing. And so in this illustration, we have an important region that is going to be important for encoding for protein, and we call those exons. And then the intervening junk regions are called introns. So here we have exon 1, exon 2 on either end of our mRNA that we're looking at with the intron in the middle. So we have assembly of proteins that come together, and as they do, they bend and twist the messenger RNA in such a way that enzymes can clip both ends of the intron away and then ligate our mRNA together and thus throw away the excess intron material, just recycle it to use it for something else. That seems like a lot of work to get rid of junk. And why would the junk be there anyway? I like to think of it like going into a battle. And our cells battle with radiation all the time in the hopes that that radiation is not going to attack a critical piece of our DNA. So if you know you're going into battle against 1,000 other soldiers, but your commander says, you know, you're the only one we got and you got to go. The general said we're going to go at daybreak. As you're standing on that hill getting ready to charge the enemy, do you have very much confidence you're going to come out of that thing alive? Unless you're Chuck Norris? No, because all those 100 or 1,000 guns, who are they going to be shooting at? Just that one, you're the target. But if you're going into battle and you've got 1,000 men against 100, your chances just went up. Just pure statistics, right? Because you've got a better chance of not being targeted when there's many more targets to choose from. The introns being in place, they are basically going to be extra hands with which to be targets for radiation damage. And so with so much junk DNA around, it's just there to protect, just from a statistical number sense, our DNA being damaged. But do you also see how, even though it's there for protection, we've got to get it out of the way before we can read the code to make a protein. So that's the splicing part. Here's another example. We have what is referred to as the primary transcript. That's your messenger RNA hot out of the ribosome. But we can't do anything with it yet. So sometimes it's referred to as pre-mRNA. You've got your 5' methyl cap, you've got your poly A tail, but we've got the introns in the middle. So we go through that splicing process. In every location we have the intron, and we get rid of them. And so what we have is our final mature, ready to be translated piece of messenger RNA. Now, sometimes, in addition to our introns, there are times that we have extra exons, the good stuff. And a great example are in your genes for your antibodies. And so as the cell and these spliceosomes, that's the name that we apply to all this conglomeration of proteins that enable you to process that messenger RNA, all those spliceosomes come together and they choose which exon to use. And they don't make, it's not like making a logical choice. It's just random. You just go in and grab stuff. And so in this case, with our typical gene that we have, the green would be one exon, and then we've got our 2 exon introns bounding that. One example of alternative splicing is you could include this middle exon, as you see here, or just completely leave it out. Here we have this little yellow piece, which would be another portion of an exon. You can see we've spliced it in such a way as you can either have it or not. So you see how we can have different transcripts depending on how we splice different parts of our pre-messenger RNA. It's just how you cut it and glue it back together. The introns and exons and the splicing, eukaryotic cells do that. Prokaryotic cells do not. So that's another difference. Polycistronic for prokaryotes, no introns for prokaryotes. Monocistronic for eukaryotic cells, but alternative splicing and introns in our pre-messenger RNA.